Hello, today we are going to talk about congenital birth defect and the reasons and the fact that I want to highlight not all congenital birth defects are due to genetic abnormalities because there are several other reasons that can manifest as congenital birth defect. Before starting the particular that topic, I would like to highlight this major organogenesis occur during the 3rd to 8th week of the embryogenesis period. So that's the importance of preconceptional folic acid supplementation that will prevent major congenital anomalies like neural tube defect. Because with the neural tube defect, those children are suffering in their entire lifespan. Not only their children but also their parents also suffering, which is a very preventable disease in most cases with preconceptional folic acid supplementation. And now we are moving to our topic, syndromic versus dysmorphic. These are not similar terms, and I would highlight and explain why. And what is meant by dysmorphic? The term dys is originated from a Greek word that is disorder, and morph means shape. So dysmorphic means disordered shape. So any congenital malformation. So I'll explain what is congenital malformation. Congenital means at birth or born with. Anomaly means abnormality. So congenital anomaly means born with abnormality. Okay. So not all congenital abnormalities are syndrome. Okay. But we can call all a dysmorphic because the shape is disordered. Okay. You get the point. Okay. Now I want to highlight what's the difference of the syndromic. To tell someone is having syndromic features, the person should have some recognizable patterns of the dysmorphic sign that have a common cause. Like Down syndrome, Turner syndrome, Marfan syndrome, we know they have recognizable patterns of the dysmorphic signs and we know the cause. To tell some child is having syndrome, we need to know the cause. Otherwise, it's only dysmorphic child. Don't label them having syndromic child. It's a different terms. When you tell the baby is having syndromic features, I expect you know the reason, you know the cause for that. Otherwise, it's only a dysmorphic child. Okay, not a syndromic child. Okay, that's the difference. Then about the birth defect. Always for the birth defect, can we blame the genes? No, because there are several other reasons other than genetic can give rise to birth defect. Let's see, those are malformation, disruptors, deformation, dysplasia, sequences and association. So as I previously explained you the difference between the dysmorphology and the dysmorphic and versus syndromic. Now I am going to explain about these terms. I know this is important for pediatric. Okay, so malformation, the term malformation we are using in places like spina bifida. There's an intrinsic development anomaly. Okay, then about the term disruptors, I have images for that. I'll go to that. Okay, here intrauterine disruptors, here amniotic band. So intrinsically the fetus has ability for the normal development, but there are amniotic band disrupting it. So it's a disruptor. The mechanism is disruptor. So the baby is born like this. So this is, we can call them dysmorphic because the morphology is different. And here we know the reason. If we know the reason, we can call it as a syndrome. So amniotic band syndrome present like this. Here, okay, we know that when we are inside our mother's womb, we are covered with amniotic fluid. So if this amniotic band is disrupting our growth, then we also can develop these features. Okay. Okay, although we had the ability for the normal development, these intrauterine disruptors disturb the development. Okay, so that they present as congenital anomaly, so we can't blame the genetic for this. Okay, then other term is deformation. For the deformation, that's also external force that alter the shape of the development. Here, when there's a severe oligohydramnion, not in this picture, and imagine when there's a severe oligohydramnion, that will alter the development of the face shape. so their phase shape is different. And then other one is sequences. For that I have image. I'll go that here in Potter sequences. In Potter sequences, there's a problem in the fetus kidney formation. So there's a renal agenesis. So they will produce less urine. That will lead to oligohydramnion. That means less fluid within the amniotic cavity. That will lead to less development of the lung. So pulmonary hyperplasia. So one step leading to other step. That will lead into other step. So that's a sequence. So here we know the reason. So that is Potter syndrome. Okay. Now we are going to next type of reasons for the birth defect, that is association. Okay. Association is there's a group of malformation occurring together that is more than expected. If it is occurring by chance, we expect it can occur in a very rarely. Okay, but here more than the frequency of the by chance, it's occurring together more and more. So that is association. But we don't know exact gene, so we don't know the exact reason. So here, rather than using the term syndrome, the best word here to use is the vectoral association. For the vectoral association, it's no need to do genetic, because we don't know the exact gene yet. Maybe in the future someone might recognize the exact gene. At that time it will be ideal to do genetic, but for the time being in 2024 still we don't know the exact gene for the vectoral association. Okay, in vectoral association what we see is vertebral anomalies, anal atresia, cardiac defect, tracheoesophageal fistula, renal anomalies and the limb defect. So lot of malformations are there, still we don't know the exact gene. Okay, we call them association. Okay, now about the other type of the term, that is dysplasia. Here the example is a thanatophoric dysplasia, other like achondroplasia, skeletal dysplasia. Here there are abnormal growth and maturation of the cell. It's a genetic problem. Okay, that's why we can't always rule out the genetic when there's a congenital birth defect, but other than the genetic there can be several other reasons. Okay, and sometimes some congenital birth defect can be purely genetic. Okay, now I'm going to move in to discuss about the syndromes. Syndromes not all are due to genetic, some are due to genetic, some can be teratogenic exposure. We know that with the rubella viral infection, that exposure, the child can get some particular type of manifestation. With uncontrolled or poorly controlled diabetes, the fetus can develop some sort of abnormalities at birth. So those are also we can categorize under the syndrome. Okay, so let's explain about the syndrome type. So some can be due to single gene disorder, some can be at the level of chromosome like Down syndrome, like trisomy 18, some due to microdeletion. Microdeletion is not a single gene but not a big chromosome, like exactly not the full chromosome but some amount of genes. Microdeletion like Prader-Willi or Angelman. Some can be polygenic like Klaproth. As I said before, some can be due to environmental teratogenic exposure, some drugs or some viruses, rubella, or sometimes poorly controlled diabetic. So as I said before, when there is a dysmorphic features recognizable collection with the known cause, we can call them a syndrome. So I think now I am at the end of the video. The things I want to highlight is not all the congenital birth defect are due to genetic. There are several other reasons, and we want to have an open mind to think about those. Okay, before directly thinking the genetic is the reason, before directly thinking go for testing for the genetic testing, first we have to think there can be other reasons also. But since karyotyping is not that costly compared to whole exome sequencing, when the child is having a lot of dysmorphic features, we can do karyotyping and we can detect some features, not submicroscopic deletion we can't detect by the karyotyping. If it is a big deletion or big translocation or any non-disjunction, that we can see in the karyotyping testing. Okay, now I am at the end of the video. I'll come with another video again. Thank you.